We'll give everyone a few moments to get logged on. Thank you for joining. Thanks for joining. We'll begin in just a moment. We want to give everyone a chance to get logged on. All right, let's go ahead and begin. Hello, everyone. Thank you for joining us for tonight's webinar as we discuss an RNA approach to rare cardiomyopathies. My name is Nikki Butler, and I'm the Director of Digital and Social Media for the Dilated Cardiomyopathy Foundation and the Genetic Cardiomyopathy Awareness Consortium. We're so glad you can be with us as we discuss this important opportunity. We have over 400 registrants for tonight's discussion. Before we begin, we do have a few quick housekeeping notes. All attendee microphones are muted so that everyone can clearly hear our presenters. We'll have a dedicated Q&A session following tonight's presentation, so please feel free to submit questions at any time using the Q&A feature in Zoom. Please note that our presenters cannot provide individual medical advice. If you have questions about your own care, we encourage you to speak with your physician. Many individuals with genetic cardiomyopathy benefit from seeing a heart failure specialist at an academic medical center. We also ask that questions remain focused on the topic of tonight's presentation so that we can make the best use of our time together. We do have a poll for tonight. Let's push that forward. All right. Are you best described as a cardiomyopathy patient, a family member or friend, a medical professional, or an industry professional? It's always interesting to see the breakdown of these results. And we had so many people interested in tonight's topic. So we appreciate you filling in your answer. I'll give you just another moment or so there. Looks like we have about 44% industry professionals, 28% medical professionals, 23% cardiomyopathy patients, and 6% family members and friends. And we'll keep rolling. I do want you to know that this webinar is being recorded and it will be posted on our YouTube channel following tonight's discussion. As registrants, you will receive an email with a link to the recording. Our YouTube channel also houses a library of our previous webinar recordings. And using the auto translate feature, those videos are available in 54 different languages. I'll begin with just a brief history. The DCM Foundation launched the Genetic Cardiomyopathy Awareness Consortium, or GCAC, in 2023 after recognizing a major gap in awareness around genetics and cardiomyopathy. We also realized these conditions couldn't be approached in silos. Many genetic mutations overlap across cardiomyopathy diagnoses, making collaboration essential. GCAC was created to bring patient organizations together to increase education, awareness, and support across all forms of genetic cardiomyopathy. In just a few years, GCAC has grown from 8 participating organizations to 11 patient advocacy groups working together alongside the DCM Foundation. That growth reflects the increasing need for education and awareness around genetic cardiomyopathy and the power of collaboration in addressing it. We're incredibly grateful to each organization represented here for being part of this effort. We're pleased to have 3 outstanding experts joining us tonight to share their knowledge and perspectives. Dr. Hussam Yunus is Chief Scientific Officer at Atrium Therapeutics and brings more than 20 years of experience in drug discovery and development, including extensive work in rare disease. He has held senior R&D leadership roles at Avidity Biosciences, NGM Biopharmaceuticals, Ionis Pharmaceuticals, and Pfizer. Chelsea Ortiz is Senior Director of Patient Advocacy and Policy at Atrium Therapeutics. She brings more than 15 years of experience in rare disease advocacy, working closely with patients, families, and advocacy organizations to ensure that patient experiences and priorities are meaningfully represented. And Dr. Quan Bui is a board-certified cardiologist at UC San Diego, specializing in advanced heart failure and genetic cardiomyopathies. He is also an assistant professor at the UC San Diego School of Medicine, where his research focuses on cardiovascular genetics and inherited cardiomyopathies. Together, our speakers bring perspectives from scientific research, patient advocacy, and clinical care as we explore emerging RNA-based approaches and what they may mean for patients and families affected by rare genetic cardiomyopathies. Thank you all for being with us tonight. Dr. Yunus, I will turn it over to you.
All right. Thank you, Nikki. Good afternoon, everyone. Really want to initially thank GCAC for inviting us today. We really look forward to sharing the important work that we're doing at Atrium Therapeutics, tackling these devastating genetic cardiomyopathy diseases. Next slide, please. All right, so first I want to start with our vision at Atrium, which is to pioneer precision RNA medicines for the heart and profoundly improve the lives of people impacted by cardiac disease. And as you can see from this vision statement, we're really aspiring to change the trajectory of these diseases, specifically around genetic cardiomyopathies, and really to make some profound improvement on people's lives, not just trying to achieve incremental improvements. Next slide. So Atrium Therapeutics is headquartered in San Diego, California. Actually, we were just founded a few months ago at the end of February of this year. We're really kind of focused on the heart and developing RNA medicines for the heart. As you can see here, we're a small but very mighty team with deep experience in rare disease. And we really look forward to continuing to partner with the patient community moving forward. We're essentially a fully functioning biotechnology company, so we do have active research and development programs, and we essentially wholly own our programs. They're all homegrown, and we have all the capabilities possible to discover and develop them moving forward. Next slide, please. Yep, thank you. As I mentioned here, we're uniquely focused on working on cardiology diseases, developing RNA therapeutics for genetic cardiomyopathies. And really, this is kind of based on our unique capability to deliver RNA medicines to the heart. And these RNA platform technology was initially designed at Avidity Biosciences, where they were really working on neuromuscular disease, and we're really applying the same technology and applying it to the heart. The strength that we have here is really around our experience in rare disease. Um, and a good number of our employees actually pioneered this platform in that neuromuscular space. Um, since we've launched in February of 2026, and in just a short few months ago, we have now been able to get our first program, which is in PRKAG2 syndrome, to be cleared. Our R&D for that program is cleared by the FDA. And Dr. Bowie will talk more about that in the next few slides. We also have, in addition to the PRKAG2 program, and a research program going after PLN cardiomyopathy, that is ATR 1086. We also have two additional undisclosed programs also focused on cardiomyopathies. Based on our experience in rare disease, we know that it's really critical to partner with the patient community, patient organizations such as GCAC, key opinion leaders in the field, and physicians. We believe that partnering is going to be critical and essential for us to achieve the goals we'd like to do and really seek to achieve the impact we really want to make. So we really look forward to partnering with this community moving forward to get your insights, your feedback, and we're going to use that information to guide our programs moving forward. Next slide. So before I go into the pipeline and the programs that we have here at Atrium, I'd really like to kind of give a background on RNA therapies, what they are, how they work in the body, and why we believe here at Atrium that this is really a new approach that we believe is going to make a substantial impact to be able to potentially treat quite a number of genetic cardiomyopathies. On the next slide. So this is a very simple schematic here that really demonstrates how the cell and tissues make all of the proteins and factors that are critical for their function. So it starts off with DNA that contains the genetic code. And the genes within the DNA essentially have the master instructions. And these are stored in every cell in the body. And those are the instructions essentially on how to make the proteins, which are the functional components and factors that allow the cells and tissues to do what they need to do to function appropriately. And RNA is essentially an intermediate copy of these instruction manuals. And RNA is essential to translate those instructions and they use the cellular processes that exist within the cell to translate that genetic code into the protein. So there's a number of diseases then that are mutations in the DNA that can change how proteins are made. And so these genetic diseases can lead to a number of things. Some of these mutations essentially could lead to a loss of a specific protein, or they could lead to just a reduced amount of that protein being formed within the cell. These are generally described as loss of function genetic mutations. Another type of genetic mutation could lead to too much of a certain protein to be made, or an aberrant type of protein that's made, or an aberrant type of mRNA that's made. Those are typically called gain-of-function mutations. And so there's different types of treatment modalities that one would be able to utilize to be able to address these different types of genetic diseases. So if you can go on the next slide. So in the case of diseases where there are mutations in the DNA, where you have loss of function, so not enough of that protein or factor, typically in this case, you use gene therapy. And essentially what gene therapy is providing that DNA code such that the cell then can make the right protein. In contrast, for those diseases where you have gain of function, i.e. too much of that protein or an aberrant or toxic form of that protein. What you really want to do here is use RNA therapy. And we believe that RNA therapy for those types of gain-of-function mutations are really the best approach. And this is, the work that we're focused on here at Atrium Therapeutics, where we specifically use what's called small interfering RNA technology. In this case, this small interfering RNA, these are very small pieces of RNA that specifically bind to what's called messenger mRNA, this kind of precursor molecule before the protein is made. And what they do is they essentially bind to the specific mRNA that's associated with this disease and degrade it. So essentially what you get with these types of therapeutics is they reduce the amount of the toxic protein or the amount of the toxic mRNA that's produced that's associated with these diseases. And the power of this approach is really to go after the genetic cause of the disease. So that's really the main utility here. So on the next slide demonstrates some of the advantages of utilizing this siRNA approach to treating a number of diseases. The first is really focused on the fact that these RNA therapeutics, and in particular siRNA, is not impacting the genetic code, so the DNA. So the DNA in this case stays intact, and you're not modulating the DNA. You're really modulating the RNA. So these molecules are fairly short-lived, and you have to continue to dose them to be able to get an effect. The second key advantage is that, as it turns out, this process around small interfering RNA and RNA silencing is a naturally occurring process. So cells use this methodology and process. And what we're essentially utilizing here is we're taking advantage of that naturally existing process to be able to now provide a therapeutic approach. So we're really utilizing an existing naturally occurring process to be able to intervene in the case of these diseases. And I mentioned this already, but the third major advantage of this approach is that we could dose these molecules in a repeated fashion. They're formulated in simple formulations. They're not viral vector formulations. So this really allows us to dose them repeatedly. And this is really important for chronic diseases because oftentimes when you dose these types of medicines, their activity wanes over time. So it's really important to have a molecule that you can dose repeatedly to be able to get long-term sustained effects. This technology is not just a scientific concept. There's actually quite a number of siRNA therapeutics and RNA therapeutics that are approved for a number of different conditions, including in rare disease. And so there's really a wealth of clinical information as well as regulatory information on these molecules for us to really be able to springboard our evaluation and assessments in the cardiomyopathy space moving forward. Right, if you can go to the next slide. So one of the limitations of the RNA therapies, and in particular siRNA therapies, is their ability to get to the right cells and the right tissues in the body where the disease exists. And if you dose these molecules as their unconjugated form or just the siRNA by itself, they're actually readily cleared from the bloodstream. So very little gets to, for example, heart tissue or muscle tissue. And so what we've done here at Atrium is that we're utilizing a new technology to be able to deliver these much more readily and much more efficiently to muscle tissue and heart tissue. And the way we do that is by conjugating these molecules, in this case here, to a monoclonal antibody. And so when we conjugate it to a monoclonal antibody, what we get is what we call here an antibody oligonucleotide conjugate, or an AOC. The way this technology works is that the antibody recognizes specific receptor on the tissue. And in this case, the receptor is called transferrin receptor 1. And it binds, and this receptor is fairly ubiquitously expressed on muscle tissue, including cardiomyocytes in the heart. And so you can think of it as kind of like a GPS where it directs that drug now to cardiac heart tissue. Once it binds to the receptor, the whole molecule gets internalized. And then this is where the oligonucleotide portion, the siRNA therapeutic, gets released into the cell, and it gets to do the actions that we're asking it to do. As I mentioned, this technology was initially pioneered at Avidity Biosciences. So we have quite a wealth of information that we know around these types of molecules that we're employing at Atrium Therapeutics for our cardiology programs. Go to the next slide. So this leads us to our pipeline here at Atrium. As I mentioned to you, our lead program, which is currently in clinical development, is called ATR 1072. This is going after PRKAG2 syndrome. The clinical trial that Dr. Bui will talk about is called Corventus, and we recently got IND clearance here in the US from the FDA, as well as in Canada. And we're excited to kick this program off. Our second program is for PLN cardiomyopathy. This program is ATR 1086. We're pushing this one forward readily as well, and we anticipate an IND filing in 2027 next year. We're also working on a couple of other programs, also in rare cardiomyopathies. And the third program here, we anticipate nominating a molecule for development also next year. So as you can see here from our pipeline, we have a lot going on for the small team that we have. At the same time, we're very excited, and there is a sense of urgency here that we have to really get these investigational medicines to clinical testing as fast as we can. Next slide, please. So before I hand it off to Dr. Bui, what I'd like to do is really kind of, if you can go back a slide, quickly describe to you how this disease manifests and how our investigational drug here, ATR 1072, works to be able to intervene in that process. So PRKAG2 is the regulatory component of an enzyme called AMP kinase. Now, this AMP kinase enzyme is a critical energy sensing enzyme in all cells, in particular the heart and cardiomyocytes. And there's a number of different mutations that can cause this disease on this PRKAG2 gene. All of these mutations essentially culminate in leading to overactivation of this enzyme AMPK. And this overactivation of AMPK leads to the disease, which is described here in terms of the hypertrophy. It can also produce arrhythmias. And one hallmark feature of this disease is the glycogen accumulation in the heart. So our therapeutic approach here with ATR 1072 is to reduce the amount of these mutant transcripts, the mutant mRNA that's formed in patients that have this disease. So we are using ATR 1072, which essentially is, again, the antibody conjugated to a siRNA. We deliver that systemically, and it gets into the heart, releases the active siRNA molecule to reduce these mutant transcripts. And ultimately, the goal of that is to get AMPK activity to normal levels. So our desire here is to get it back to normal. And the hypothesis here is that in doing so, we're going to relieve the genetic cause of this disease, reducing the amount of glycogen that is deposited within the heart that leads to a number of these symptoms, as well as improve the arrhythmia burden. So with that, I'll hand it off to Dr. Bowie to talk about our clinical trial.
All right. Thanks, Dr. Yunus. I'm excited to be here again. My name is Dr. Quan Bowie. I'm an advanced heart failure and transplant cardiologist at UCSD, and I have a true passion really for genetic cardiomyopathies. And thank you for Greg Ruff and the GCAC team and Atrium for this opportunity. I do want to say I am not an employee or paid by Atrium Pharmaceuticals, and I want to be able to give unbiased opinion on RNA therapeutics. But I am a principal investigator for the clinical trial that I'll discuss later in this presentation. So let's dive into PRKAG2 syndrome. Next slide. So PRKAG2 is a rare genetic cardiomyopathy. And as people know, cardiomyopathy is a disorder of the heart muscle. And in this case, you have a mutation in PRKAG2. And PRKAG2, as Dr. Yunus mentioned, is really responsible for regulating the energy inside the heart. And when this mutation occurs, you have this overactive signal that occurs, and you have glycogen, which is a form of energy that builds up inside the heart muscle. And when it builds up in the heart muscle, it can cause thickening of the heart. It can cause abnormalities related to the electrical conduction system, and ultimately lead to heart failure and abnormal heart rhythms. And our biggest fear is a risk of sudden cardiac death. In terms of the way it's inherited in an autosomal dominant fashion. What that means is that it tends to affect generations of families. And so if a person or a patient has the mutation, then children will have a 50-50 chance of having that same genetic variant. And up to 100%, nearly everyone with the genetic mutation that we know at this time will develop symptoms. And so this is really important to identify people as early as possible so that we can potentially provide early treatments and prevent irreversible potential damage. It's a really rare disease. About 1,000 to 2,000 patients are estimated to have PRKAG2 syndrome in the United States. And there are no approved therapies that treat the underlying cause of disease, right? And so, again, we do our best to manage patients with heart failure and manage their arrhythmias with the medications we have, but there are no approved therapies that treat the underlying root cause of disease. Next slide. And so how can PRKG2 syndrome show up? As I mentioned before, due to glycogen buildup within the heart muscle, it can cause heart thickening. So you can see thickening of the heart muscle, and it can look very similar to other diseases like hypertrophic cardiomyopathy or cardiac amyloidosis or Fabry disease. And genetic testing obviously can help discriminate, between these different types of cardiomyopathies. And ultimately, due to this abnormal thickening and glycogen buildup, you can develop heart failure. And so patients can develop symptoms of shortness of breath, fluid retention, and dyspnea on exertion, trouble breathing when they exercise. The other manifestations include electrical conduction problems. And so due to the electrical wiring issues, some patients may need a pacemaker at a young age. It can also cause a short circuit of the electrical system and cause abnormal heart rhythms like Wolff-Parkinson-White, which can be life-threatening. It can also cause other arrhythmia issues like atrial fibrillation and flutter, and also ventricular arrhythmias like ventricular tachycardia and fibrillation that increase the risk of sudden cardiac death. And so I wanted to turn over to the next, the right-hand side of the screen here that shows some of the expected morbidity and mortality issues that patients with PRKG2 patients can experience. And I will give a caveat that these are based on estimates, are based on small studies of patients with PRKG2, but kind of highlights what patients may ultimately face in the future. So about 20% of patients will experience a sudden cardiac arrest by age 40. About 50% of patients may have a major heart event by age 50. A handful of patients will need a pacemaker or defibrillator by age 40, and approximately 70% will develop this pre-excitation or the short electrical shortcut that leads to abnormal heart rhythms by the age of 40. So you can see here, this is a genetic cardiomyopathy that causes a lot of morbidity and impacts patients' quality and quantity of life. Next slide. So next, I'll talk about the Corventis phase 1/2 trial. Again, it's the first clinical trial to target PRKG2 syndrome. But before I go into details about the trial, I wanted to give you all maybe the appropriate framing to set this up. So I always like to think about where the state of therapeutics are in heart failure and compare it to oncology. And it kind of helps us understand where we are currently and where we are going in the future. So in cardiology and heart failure specifically, we tend to treat patients once they develop symptoms. And it's really based on the underlying heart failure or cardiomyopathy you may have. So if you have a dilated cardiomyopathy, a weak heart muscle function, if you have preserved ejection fraction or hypertrophic cardiomyopathy, we have therapies that kind of treat those symptoms and underlying pathophysiology. Compared to oncology and cancer, there's a different mindset, at least in medicine, where we treat patients once we see the disease, once we detect it, even in the absence of symptoms. And therapeutics are really based on the underlying genetics of the disease. And that's kind of where we're going in precision cardiology. And I'm really hopeful that will be able to target the underlying genetic and molecular mechanisms of cardiomyopathy in the near future clinically. So next slide. So Corventis, again, is testing ATR 1072, which Dr. Muniz explained is a RNA-based therapy for PRKG2 syndrome. It is a given every 6 weeks IV infusion. And in this clinical trial, there's two parts of it. There's a part A and a part B. And the goal of the overall trial is to make sure that we can find the right dose of the medication and also to ensure that it is safe. So in part A, there are 3 different cohorts with 4 patients each. And you can see here, there is escalation of the doses from 40 milligrams to 160 milligrams. And the patients will be ultimately followed for 48 weeks. And then part B, which is a larger part of the cohort, and there will be 25 patients, and they'll be, again, followed for 48 weeks. And then that will roll over into a long-term extension. It's a open label adaptive trial design, so it's not, no one in the trial will get placebo. Everyone will receive the drug. And 37 patients are to be enrolled at 15 sites globally. And they're looking for adults aged 18 to 65 with the PRKG2 mutation and evidence of cardiac manifestations. The primary endpoint, as I mentioned, is related to safety. And they will be looking at secondary endpoints to see how effective their siRNA therapy is. So in part A, patients will require a endomyocardial biopsy, a heart biopsy, so that they can look at mRNA and siRNA tissue concentrations. But that's only for part A, and they do not expect that to be needed for part B. Some of the exploratory endpoints will include echo and other imaging parameters, as well as different lab tests and cardiac biomarkers. Next slide. And so who can participate in the trial and what to expect? As I mentioned before, they're looking for adults age 18 to 65 with PRKG2 with cardiac manifestations. And, the best way we'll talk about this on the next slide, the best way to see if you might be a candidate is to reach out to a study site team like us here at UCSD, and we can review your medical records and see if you might be eligible. If that's the case, we can then have you come over for screening, and then you can, if you pass the screening, participate in the clinical trial, which includes a treatment period for 48 weeks, and then a long-term extension for up to 42 weeks. And afterwards, you'll be followed too as well to make sure that we're checking different health-related measures and safety for that duration. So again, this is a brief summary of the Corventis trial and a brief summary of PRKG2 syndrome. And I'm happy to answer any questions in the Q&A. And I'm going to hand it off to Chelsea to wrap this up.
Great. Thank you, Dr. Bowie. As we wrap up, I just want to thank everyone for spending time with us today, and especially thank GCAC for creating the opportunity for us to have this conversation with the community. At Atrium, we really believe that developing medicines for rare diseases has to happen in partnership with the people and the families who live with these conditions every day. That means listening, learning, and making sure that the patient and caregiver perspective continues to inform how we think about our work. As you can see on this slide, there are several ways Atrium engages and supports the patient community. We continue to build on our meaningful partnerships with our patient advocacy organizations and advocates finding ways to collaborate and support the community needs. We also have launched a Corventis clinical trial website for our phase 1/2 clinical trial. This is a patient-friendly resource for patients, caregivers, families, and even healthcare providers to learn more about the trial. Please note that this trial is not yet opened for enrollment. However, we are working hard on our end and hope to have enrollment opened in the upcoming weeks. If you're interested in learning more, you can visit corventistrial.com. There is also an ongoing natural history study in PRKAG2 syndrome. Natural history studies are extremely important for researchers to better understand the disease and how these conditions progress over time. If you're interested in learning more or would like to contribute, you can visit citizenshealth.com. And most importantly, we want you to know that our door is open. If you have any questions, feedback, or simply want to connect with our team, you can reach out to us at patients@atrium-tx.com. We know that there is still so much to learn, and we're incredibly grateful to this community for helping us learn it. So thank you again. And with that, I can pass it back over to GCAC.
Thanks so much, Chelsea, Dr. Eunice, and Dr. Bowie. Thanks for your presentation. And now is the Q&A. And so if you have questions, would love for you to go in the question area and ask the questions away. Only you will not be able to see your question. Only the presenters will actually be able to see the question. Number two, I'd like to thank Atrium for, doing what they do, all the dedicated people there. As the executive director of the DCM Foundation and GCAC, a genetic cardiomyopathy patient, heart transplant patient, I realize how important it is to have organizations like you that are creating potential new therapies in our area. I think it gives hope to all the patient populations. And then the third thing I'd like to say before we get into the Q&A is if you have PRKAG2 or the PLN gene, it would be great if you could reach out. Doesn't mean you are interested in their clinical trial in the future, either one that's coming out, but please reach out to patients at atrium-tx.com. I always say it's important for patients to get involved and to investigate any new potential therapy. Doesn't mean you have to do it, but we really encourage you to investigate and learn more about it. Great. Again, we're getting some questions in, so we'd love for you to ask away. For Dr. Eunice, question for you: what exactly is siRNA, and how is it different from gene therapy?
Yeah, happy to answer that. So siRNA, it stands for small interfering RNA. Again, this is kind of an endogenous mechanism that's used in cells in the body. Essentially, it's a small piece of RNA. Typically, these are 19 to 21 sequence length pieces. This is a double-stranded RNA molecule. And that's very different than gene therapy, right? The purpose of the siRNA molecules is to interact with the mRNA and destroy the target mRNA. Gene therapy is really focused on introducing a DNA and producing that genetic code to make protein, to make mRNA, to make the protein. So they're very distinct. And as I mentioned in the presentation, you have to apply these to different types of diseases. So if the disease is caused by lack of a particular protein, the best way to address those would be with gene therapy. In the case of what we're doing here at Atrium, we're really focused on these gain-of-function type mutations, where we believe that the siRNA platform is really a powerful approach, a new approach in this space.
Great. Thank you. For Dr. Bowie, question: what should patients ask their cardiologist if they think their cardiomyopathy may have a genetic cause?
Yeah, it's a good question. I think that lots of cardiologists, unfortunately, don't know a lot about genetics at this time, and it takes, so much is evolving, and we're not supporting our cardiologists with the right tools and resources to help them treat these patients. But I think the first step is to ask them for a genetic test, right? That's the first step to see if you have a genetic cardiomyopathy, and we know that we're only testing 1% of non-ischemic cardiomyopathies across the country. And so if you are talking to your cardiologist and you ask them for genetic tests and they say, sorry, I can't do it, Greg, and you and your organization has the resources and tools to get people the appropriate testing. So that's the first thing I would do. And then connecting with different cardiologists across the country who have a genetic focus could be another avenue to explore.
Great, great. No, thank you very much. And again, any of the patients that are out there, if you suspect that there's multiple people with cardiomyopathy, suspect genetic, feel free to reach out to us. Ask your cardiologist. Unfortunately, a lot of cardiologists will say you don't need tested. That's not true. But feel free to reach out to us, info@dcmfoundation.org, and we're happy to answer your questions and maybe get you along on the right path. Dr. Yunus, is the same mRNA, is this the same mRNA as the COVID vaccine and the recent approved flu vaccine, or is it different?
Yeah, this is different than the COVID vaccine or the flu vaccine. So in the cases of those vaccines, what you're actually administering is the mRNA, that kind of messenger mRNA intermediate that goes from DNA to RNA to protein. That's what you're dosing. In the case of these RNA therapies that we're focused on at Atrium, these are small interfering RNA, so very, very different types of molecules.
Great, great. One quick question. When will the Corventus trial begin enrolling? Either one, I don't know, Dr. Yunus or Bowie, which one? So do you
Yeah.
have a time frame when it will start enrolling?
Yeah, so currently we're have identified the investigators and the sites that will assist in setting up the trial. As those get initiated, we anticipate enrolling by the end of the year and dosing our first patient by the end of the year.
Okay, perfect. Thank you. Thank you. Here's a question. I'm going to try to answer it, but obviously either of you can answer it too. First question is there any research in the pipeline on RNA treatments for loss of function defects, i.e. desmoplasmin cardiomyopathy? I do know of some research, that is actually happening on this. And Christina, you'd asked the question if you want to reach out to me individually at info@dcmfoundation.org. I'm happy to have a further discussion with you on that. So another question from Sanjay. As you expand the pipeline beyond PRKAG2 and PLN, where do you see the biggest gaps in the preclinical translational package, particularly cardiac disease models, PKPD, biomarkers, or genotype-specific efficacy models? And Sanjay, thanks for the question. I have no idea what you're asking, but hopefully Dr. Yunus or Bowie do.
Yeah, I can try to tackle that, and really appreciate Dr. Bowie also chiming in. These genetic cardiomyopathies oftentimes don't have animal models. And so we spend quite a bit of time, certainly at Atrium, trying to think through creating these animal models, evaluating them. And the premise here is that we're using really human genetics to substantiate the rationale. And one of the powerful aspects of the RNA platform that we have is that we can deliver these molecules to the heart based on our preclinical data, and we're really kind of going after the root cause. So we really have a lot of conviction around the human genetics. The animal models are very helpful. They guide us in many different ways in terms of evaluating biomarkers, getting us to kind of focus on certain aspects of the disease. So that they are important, but equally important, I think, are the human genetics and our conviction around going after these genetic cardiomyopathies.
Okay, great, thank you. And so here's a statement in the question. Thanks for the presentations. I am Dr. I'm going to butcher your name, Girkov from Buenos Aires, Argentina. Hey, thanks for joining us from Argentina. And their question is, will you accept international patients in the clinical trial?
Yeah, at this point, we've gotten IND clearance here in the US, as well as Health Canada has cleared the clinical trial. So really focused here in North America at this point in time.
Okay, thank you. Thank you. And I would say stay tuned as the trials expand, with different information, stay tuned. Again, if you want to reach out to info@dcmfoundation.org, happy to give you updated information as things progress with this. Thank you. Sue asks, will patients outside of 18 to 65 age range ever be accepted into the clinical trial?
So I can't speak on behalf of the company, but I have asked Atrium before, because we have some pediatric patients who are affected with PRKAG2 syndrome. So I think at this time they're hoping to keep it age 18 to 65, but they have told me that they are exploring options to potentially expand that in the future. Correct me if I'm wrong, Dr. Yunus.
Yeah, no, that's correct. It's really important for these first in human type trials to understand the safety of the molecule. So, from that perspective, we believe that going after this 18 to 65 age population really gives us the best handle on understanding the safety, at which point, we can consider expanding outside of that range.
Yeah, great. Thank you. Alexi has a question. Can you please comment on the dosage? What is the milligrams of AOC and siRNA?
Yeah, so the doses that were presented by Dr. Bowie, those are representative of the siRNA dose, not of the entire AOC molecule. So it represents flat dosing. So each patient, regardless of their weight, is going to get that absolute dose of the siRNA.
Okay, great. Thank you. Doug, you have a really good question. It's more of a business question. So again, if you want to reach out to me at info@dcmfoundation.org, I'm happy to put you in contact with the business people at Atrium to answer that question. John has asked, is it possible a version of the approach described here could benefit DSP cardiomyopathy patients? And I don't know if Dr. Bowie, you want to take this. I know Dr. Yushi probably, or either one of you, can you answer that?
So I think, Greg, you kind of mentioned that there might be some preclinical work here, right? But as of now, based on what I know, and maybe Dr. Yunus can expand on other potential possibilities, there isn't a way, at least now that I know, where you can use an RNA-based therapeutic for a predominantly loss of function DSP-related cardiomyopathy. But I do know people are trying to work on this, different specific ways, but I think it's still very preliminary.
Very happy. Yeah. And if you are not in our DSP Facebook group, please join it. It's at geneticcardiomyopathy.org. And there's, I think, 600-700 members, and it's a lot of great place to get information, to get support. Are lamin-related diseases a candidate for RNA delivery treatments?
That's a good question. The only therapy I know right now that's in clinical trial is the Novocor, right? It's the AAV gene therapy. I haven't come across any particular company looking at developing an RNA-based therapy for Lamin. And obviously, it comes down to the genetics. Some of these RNA-based therapies are, again, what Dr. Yunus mentioned, best for gain-of-function mutations, specifically for siRNA. So doesn't mean it's not possible, but right now I don't know of any at this time.
Yeah. I'm a laminate patient myself, family members. And so I do know that there are dozens of companies working on both AAV and RNA therapies right now in the pipeline. And so there is a lot development. It's happening, pretty quickly. And so again, I would stay tuned to our website. If you haven't signed up for our emails, we'll announce more and more that's happening in the future. Can't say exactly what it is, but there is a lot happening in this space, and it's very exciting. I think for all patients in this space. So Anonymous says, I have an ultra rare variant that led to cardiomyopathy, heart failure, and heart transplant. Welcome to the club. How do you find individuals with the variant you want to research? How many cases of a variant are needed to encourage researchers to learn more about it? And so maybe Kwan, that's probably good for you. You're a researcher in this.
Yeah, I mean, I think as a researcher and also as an advanced heart failure transplant cardiologist, I think the first step is you don't know how rare it is, right? We're not doing enough genetic testing, so we may think something may affect only a certain number of individuals, but when we do more testing, we might find out that there might be actually more. But I will say that, we're interested as at least researchers and cardiologists about just developing therapies that will improve the care of our patients. My mentor, Dr. Eric Adler, he was he pioneered the gene therapy for Danon disease, which is a really rare genetic cardiomyopathy, right? And it only affects potentially thousands of individuals across the world. So, and that has been a very successful gene therapy clinical trial, at least in the first phase 1. So again, we're don't be discouraged if you think you have an ultra rare variant. Just reach out to organizations like GCAC and Greg, and Greg can help connect you with other like-minded patients and researchers like me who are interested in developing new therapies and improving the care of genetic cardiomyopathy patients.
Yeah, please reach out. In my view, we're working really hard to identify more patients. It seems like the research in this area and the drug therapy development in this area is outpacing the known patients. And so we're working hard to identify patients for researchers and potential drug developers. And then we're also working hard to increase genetic testing. Thank you. Marco asks, is the delivery mechanism with the antibody safe in humans regarding peripheral iron metabolism? And are there any known off-target mechanisms? So that's definitely not a patient.
Yeah, happy to address this one. So, yeah, as I mentioned, the antibody does interact transiently with the TFR1, which is critical for iron uptake into cells. We have based on the programs at Avidity Biosciences where they've evaluated the same platform in rare disease and neuromuscular space. We have a good handle on the safety profile of these types of molecules. So, all of that know-how, we're applying it here at Atrium Biosciences. So we really have a good way of being able to safely investigate these molecules in patients moving forward.
Great. Thanks. And Marco's part 2 question, you may have answered part of it, but are you also targeting autosomal dominant diseases in which loss of function is the mechanism? Can you see this delivery system to deliver mRNA to increase the levels of a given protein as in ARVC, for example?
Yeah, I mean, this is an interesting concept, right? At this point, we're really focused in on these, gain of function type diseases because we believe that's really where, the greatest efficiency is around this platform technology. The the mRNA therapies, just like the COVID type vaccines, right? They're very different. They're much larger molecules, so you need much more complicated formulations to try to deliver these. This is not something that we've ventured into at this point.
Great, thanks. Mark has a question. Is there a risk of loss of physiologic AMPK function with this therapy?
Yeah, so this is something we've investigated quite closely. Before you get IND clearance, you have to do a number of safety studies in animals, and we've been able to robustly reduce the G2 mRNA substantially to give us some really good comfort around that. In terms of the pathway itself, there are other isotypes, in particular G1, which is another isoform in the pathway. So there's some redundancy that's built in. We don't really believe, based on our preclinical data, that we need to substantially reduce G2 because the disease is caused by overactivation of AMPK. Our goal is to really get that back to normal levels, not to reduce it below normal.
Very good. Jody has a question. Have there been other areas of study, or maybe what she says, have there been other RNA therapies developed in different disease states that have successfully passed clinical trials without significant adverse effects?
Yeah, I think that's the one that I think about is cardiac amyloidosis, specifically ATTR, right? So that's, patisiran, which is an siRNA therapy, has been successfully seen in clinical trials to be effective for patients with ATTR cardiac amyloidosis, right? And that's using a different delivery vehicle, not an antibody conjugate, but lipid nanoparticles, right? And so there have been siRNA therapies being used, antisense oligonucleotide therapies, with different delivery vehicles. And so, yes, they have been shown to be effective in clinical trials and safe.
Great. Thank you. We're getting close to time. If I think there's a couple around, about other genes. Will you do something similar in the near future? I think one of the things is I know you have 2 undisclosed targets for rare genetic cardiomyopathy. I think you mentioned, and I, again, there are a number of companies that are working on potential therapies. So stay tuned. Question, is there any chance of Atrium's platform being made available for development of ultra-rare disease siRNA drugs in the future? Can this platform that you've developed be used in other areas with other companies, I guess?
Yeah, I mean, at this point, we're really focused on working on the programs that we have. Certainly, I think what we've realized is that there's really a lot of opportunity to use this platform. Given that we could deliver now this powerful modality of siRNA and RNA type therapeutics to the heart, it really opens up a lot of space. So we really, look forward to working with the community and partner, with folks that have ideas about how we could ramp up this work.
Okay, great. I think the other questions have been answered already. Again, very much appreciate Dr. Yunus, Dr. Bowie, Chelsea, Nikki. Thanks all of you for attending, and I'll turn it over to Nikki.
Thank you.
Thank you.
Well, thank you all for being here and for taking the time to share and to learn more about this important research. As I stated earlier, registrants will receive an email with a recording of tonight's webinar. And this email will also include Atrium's contact email, patients@atrium-tx.com, and it will include a link to register for our next webinar. Our next webinar will be held Wednesday, September 9th at 6 p.m. Eastern Time. Our topic will be Lam-A gene therapy, a new approach to Lam-A cardiomyopathy. Registration is open and information can be found on the DCM Foundation and GCAC websites. Remember that even if you cannot attend live, please register and you'll receive a copy of the recording via email. Thank you to all of our sponsors whose logos you see on the screen. Their support allows us to present this important information. And thank you again to all of the patient partner groups for their ongoing support. That brings us to the end of tonight's webinar. I'd like to thank Chelsea, Dr. Eunice, and Dr. Bowie for sharing their time and expertise with us and for helping us better understand the exciting research taking place in genetic cardiomyopathy. And thank you to everyone who joined us this evening. We hope you found tonight's discussion informative and helpful. Please continue to stay connected to the DCM Foundation and GCAC for information about future webinars, research updates, clinical trials, and resources for patients and families. Have a wonderful evening, be well, and we'll see you in September.